Welcome to the CannMed Coffee Talk podcast, where we talk with the leading minds in cannabis science, medicine, cultivation, and safety testing. I am your host, Ben Amaralt. I am the Senior Marketing Manager at Medicinal Genomics and proud member of the team that puts on the CannMed Summit. The CannMed early bird deadline is fast approaching, which means prices will jump $500 for all of our ticket packages on February 20th. So, if you are planning on joining us in Puerto Rico this June, don't wait. Buy your ticket today and enjoy those savings. And if you're still on the fence, I invite you to go to CannMedEvents.com and check out our speaker lineup, our schedule, and the unique networking events we have put together to connect you with the leaders who are shaping the industry. Just a heads up, we will plan to announce details about our Cannabis Genomics Workshop later this week, which is going to be one of the greatest collections of cannabis genomics experts ever assembled. If you are at all interested or curious about how genomic sequencing can be used to create novel varieties, protect IP, and breed for pest and pathogen resistance, this is a workshop you don't want to miss. Sign up for email alerts at CannMedEvents.com to make sure you do not miss that and all the other announcements. Our guest today is Dr. Punya Nachappa. Punya is an associate professor at Colorado State University in the Department of Agricultural Biology. Her research program focuses on understanding the interactions between plants and insect vectors to manage plant pests and diseases in hemp. At CannMed 25, Punya will present Biology and Management of Hop Latent Viroid, which will describe the impact of HLVd on hemp yield and cannabinoids, explain how thrips and aphids transmit the viroid, and evaluate the efficacy of using chemical elicitors to control HLVd infections. During our conversation, we discussed how Hop Latent Viroid has been reported in every hemp-grown state in the US and Canada, how HLVd spreads through infected clones, mechanical contact, seeds, and insect vectors like aphids and thrips, possible reasons why hemp varieties appear more resistant than cannabis varieties, including genetic diversity and environmental factors, the trade-offs associated with using chemical elicitors on infected plants, and strategies for preventing HLVd spread, including strict sanitation, insect monitoring, frequent testing, and breeding for genetic resistance. Before we get to my conversation with Punya, I'd like to thank this episode's sponsor, Advanced Nutrients. Founded in 1999, Advanced Nutrients was the first to develop a complete nutrient system that unlocks the true genetic potential of the cannabis plant. Since its inception, the brand has introduced more than 50 innovations to the cultivation community and continues to revolutionize the space through proprietary scientific discoveries. Learn more at advancednutrients.com. Okay, and without any further ado, please enjoy my conversation with Dr. Punya Nachappa. Good morning, Punya. Thank you so much for joining me on the podcast.
Thank you, Ben. It's been several years that I did my last podcast. So I'm happy to be back here talking about another important pathogen in the cannabis and hemp industry.
Yeah, that's right. You were on the podcast before talking about beet curly top virus in hemp. I'll put a link to the show in the show description so people can check that out. And I remember after we did that, I was saying that you need to come down to CannMed, and that year it didn't work out, but we're excited that you're going to be in Puerto Rico this year, giving a talk on one of our favorite topics, Hop Latent Viroid. If people have been listening to the podcast for a while, we've done a number of different talks on it. It's an emerging problem in the cannabis space, and it always gets a lot of viewers and listeners because it is so interesting. So I'm excited for you to share some new information that you've learned about the viroid and that you'll be sharing at CannMed 25. But before we get into the new stuff, I was hoping for those new listeners or new viewers, you could provide an overview of Hop Latent Viroid and explain its significance to the hemp and cannabis industry.
Yes, will do. As you mentioned, Hop Latent Viroid is an emerging pathogen, but probably doesn't need much introduction because it's just spread like wildfire. No pun intended, LA is battling these fires, but it's been such a bane to the cannabis industry. Originally, as the name suggests, it was identified in hops. That's the first host, but very quickly jumped from hops to the nearest relative of hops, cannabis, and seems to be having a more severe impact in cannabis than it did in hops, where it did affect the quality, but in cannabis, it seems to have more significant impact. I think it was identified, or at least published reports was in 2019, but maybe the industry had an idea of what was happening a couple of years before that. So the first reports came out in 2019 in cannabis, and then my lab subsequently found it in our field survey of hemp here in Colorado in 2021. And now pretty much every hemp growing state has reported Hop Latent Viroid. So it's spread all the way New Mexico up to Washington, and Canada has also been, ravaged by this disease. I think I saw somewhere that the reported impact was $4 billion annually to the cannabis industry, which is staggering because it impacts the flower really badly, right? Causing that so-called dudding, where the flower, it impacts the trichomes, and that's where the cannabinoids are made. So CBD and THC levels are reduced. And not only that, the biomass, the plant is stunted. There are issues with the quality of the plant, all in all impacting economics and downstream pharmacological applications as well. So all this is well characterized in cannabis, but in hemp, right, with the THC legally classified as cannabis sativa with THC levels of less than 0.3% dry weight, we did not know the actual impact. Although field studies have shown that we can report it or identify it in field-grown hemp. So that was one of the research areas that we focused on in the last couple of years, to do a systematic study to understand the impact on field-grown hemp.
Yeah, no, I've been very aware of the problem in the cannabis industry, and of course, growing indoors, growing from clone, and how that's a great way for HLVd to spread. I wasn't quite aware that it was being seen in outdoor hemp.
All of the reports from these other states, at least, in the universities, we're not allowed to work on cannabis. So nothing over 0.3% THC. In fact, at the Department of Agriculture here at Colorado State, at Colorado CDA, would periodically come and test our plants to make sure we are under the legal limit and the plants don't go hot. So all of the reports from these different states were made in field-grown hemp, except the ones from California. The initial ones came from cannabis. So we are finding it in field-grown hemp. Now, we just published, it's not out yet, what we call a metagenome or a metavirome. So we're surveying hemp, send it out for sequencing, and then try to identify the viruses. In the last one we did, this was 2022, 23, we did not find Hop Latent Viroid in field-grown hemp, which is good news, at least for the hemp growers. But there's a whole nother suite of new viruses that we've found, but not Hop Latent Viroid, which we did back in 2021.
Oh, interesting. So you found it in 2021, but your more recent surveys that you're not finding it.
Not finding it yet.
Interesting.
Yeah. Yeah.
Any ideas where it went?
Good question. Now, this might segue into, some of the data that I'll be presenting in CannMed, where we did a field study to see the impact of Hop Latent Viroid on hemp. And guess what? The infection rate was very low in hemp. It was really difficult to get hemp infected with Hop Latent Viroid. It's supposedly highly transmissible, right? It's the TMV, tobacco mosaic virus, which is a highly transmissible virus. It's often called the TMV of the viroid world, but we couldn't get our plants infected. I think it might have to do with the genetic diversity of hemp as opposed to the clonally propagated, hydroponically grown cannabis. That could be one reason. So I mean, this field, well, it's kind of a field. We call it a cravo. So it can be partly covered, or you keep it open. So I would say it's a hybrid between a greenhouse and a field. But you plant the hemp in the soil, so it can be considered field-grown hemp. So in this study, the plants look just great. I mean, we may have gotten like 10% infection rate. So I feel like the healthy environment, the genetics of hemp made it just outgrow or outcompete the virus. Then the other aspect is, we do these surveys. We're just going into our fields, whatever variety the grower has, we're sampling that. But it could be that certain varieties of hemp are more susceptible and some are not. So there's so much left, to be researched. So we were very surprised not to find any Hop Latent Viroid in our second metagenome.
Interesting. And what tissues were you testing, if I may ask?
A good question. So we always do the leaves. With hemp, the roots, and I know in cannabis you often do the roots, which again, because of the growing conditions, it's easier. With hemp, the roots are much harder to extract from. So we test the apical leaves, but we also sample from different parts of the plant because we are seeing that, and this has already been known, some of Dr. Punja's work has shown it's not equally distributed in all parts of the plant.
Sure.
So we are very confident about our sampling, and we also test the stem. Didn't find it, and metagenomics is a highly sensitive technique, right? You could get a few reads, right? And we detected 0. So
interesting. Yeah, and I know that some research and some of the research that our CSO has done here with, purposely inoculating some plants, the viroid will go down into the roots and sometimes stay there. And like you said, it never kind of progresses up into the later stages of the plant, and it goes through flower and it looks great. If you test the roots, it's there, but it never really emerges higher. So it could be something like that.
It could be something like that in the hemp too. We were thinking about actually testing the soil that we did our field study in.
Yeah.
And that's something we are planning to do.
Interesting. So one of my questions were, what were some of your most surprising findings in regards to your study? I think that's a very surprising finding. Was there anything else?
No, well, once we were, my former master student, he just graduated, Luke Dial did this work. When we were going out there, I mean, he just lived there in that cravo. We knew that this is, we're not going to see any significant changes in any of the yield parameters. Cannabinoids, we were still unsure. But then we decided to separate out the symptomatic. So we did have a few symptomatic plants. When we separated out the symptomatic plants and the asymptomatic plants, and both were HLVd positive by PCR, but when we did that, we saw a reduction in CBD and THC levels. So it's a little bit more nuanced than just positive and negative. So growers have to really keep an eye out for the symptoms, because the asymptomatic plants, like you said, just did fine.
Mm-hmm.
But they
Yeah,
are indeed positive. So that was an interesting finding, which I think others are also figuring it out. But so many different things, so many things were different between our study and what's happening in cannabis. Uh, um, and this is not published data, and I don't know if I could present this in CannMed. We did a screen of 30 hemp genotypes that we got from one of our collaborators here. It's a hemp genetics company called New West Genetics that we work closely with here in Port Collins, and we do see some genetic diversity, some variation in Hop Latent Viroid levels. So all of this was exciting and new data for us.
excellent. No, and in reading through your abstract as well, it said that you found that cannabis aphids and thrips can acquire and transmit HLVd as well. Can you talk a bit more about the significance of that discovery?
Yes. So this was, it was not surprising. In fact, what was surprising was that in hops, they found that hops aphid did not transmit hop latent viroid. And this was published where in our world, I mean, aphids, as you can see in my background, see how prolific they are on hemp, and they can pretty much and they've been shown to transmit other viroids. So it was kind of surprising that they didn't find it in hops, and so we set out to test aphids and thrips. And the reason we picked those are they are the most common greenhouse pests, right? They love the greenhouse environment because it's hot, humid. This makes the populations grow faster. And there's a year-round supply of host plants for them. Within 5 minutes, both insects can acquire the viroid, which is really interesting. It is in the phloem, and aphids feed in the phloem of the plant, but it's also but thrips have much shorter mouth parts. Aphids' mouth parts are like mosquitoes. They're the mosquitoes of the plant world. So they can go all the way down to the phloem and suck up the viroid. Thrips are not, but they must be picking it up from their cell content feeders. They're going to just suck up all the cell content, cytoplasm, nucleus, all of it. So there are different mechanisms by which they're acquiring the viroid, but both are doing it super quickly. And so that's great. Any insect could acquire it, but the proof of the concept was to then test transmission. And to do that, we then starve the insects to make sure all of the gut content is out of the insect. It's not just in there transiently, right? And then we place those insects on healthy plants, and sure enough, both of them can transmit. I think my other master student is retesting some things, but retesting some assays, but it's between 10 to 25% transmission. And we sequence every single amplicon. We don't just rely on the band, on the PCR gel band. Every single one is sequence. So not entirely surprising. The other one that I'm very keen to test is spider mites, which is another huge problem, a huge greenhouse pest. And we also find it in outdoor hemp. So a couple of other insects to test. The other interesting aspect is last year, we saw that hops can also infect other hosts, right? Some really important host plants like tomatoes and chrysanthemums. So we want to see if these vectors, cannabis aphids or thrips, can transmit it to other host plants as well. Because it just takes a few minutes to acquire and seemingly a few minutes to transmit too.
Interesting. No, and what about, I remember from our last conversation, the leaf hoppers, because I know that those transmit the beet curly top virus, correct?
We've not thought about leaf hoppers, but I'm going to make a note. Yeah, they are not a huge greenhouse pest, and they need, so that's the bit. So, but, HLVd is outside. So I would hypothesize because they are phloem feeders, there's no reason why they would not be able to do it. But the level of the efficiency um, would be different. Look at you, Ben. You are a budding entomologist.
I just pay attention, I guess. And another thing that you brought up, the significance of starving the insects before you kind of test the transmission. So, explain kind of the reason that's important.
So, when insects consume the plant sap or, they chew the leaves like a caterpillar would,
Hmm.
whatever is in the leaf is in the caterpillar or in the aphid. If you were to grind it up and test it, they would be positive for
I
X,
see.
let's say Hop Latent Viroid. That doesn't mean they can transmit it because once they poop it out, so goes the pathogen with the plant material. Does that
Hmm.
make sense?
Yep.
So to be called a vector, the virus or the viroid have to bind to the insect mouthpart, to the stylet, and then bind in the gut, and then bind and again find the salivary glands and come out through that mechanism, not just be pooped or
Mm.
do it transiently. And to do that, we allow like a 2 to 4 hour starvation period because it's shown to get rid of all the gut contents, and then you go ahead and you do your assays.
Interesting. Okay. So it's not that it's just kind of taking infected
Right.
phloem or sap from one
Correct.
plant and depositing it elsewhere. It's
Yes.
actually systemic within the insect.
Exactly. That's a great way to say it. So the way the process works is insects pick it up, it attaches to the stylet. Stylet is the mouth part, goes into the gut. Then the viroid will then come out of the gut into the body cavity, where the insects have something called hemolymph, which is the blood of the insect,
Mm.
and then reach the salivary glands, get into the salivary glands, and when the insect feeds on a healthy plant through the saliva, the viroid has to come out. This whole mode of transmission has to happen for it to be called a vector.
Interesting. Okay. All right. Very cool.
The
Um,
one thing I just wanted to add, one thing I
sure.
wanted, so we've been doing acquisition assays, how quickly they can acquire it, how quickly they can transmit it. But what we want to know, like I told you, this mode of transmission within the insect, we are doing some confocal microscopy where we tag the viroid with GFP, the green fluorescence protein that can glow
Hmm.
and show us all the different parts of the insect body that the viroid is moving through. So we get a good understanding of how it's circulating systemically in the insect.
Oh, wow. Very cool.
Yes, I'm so excited.
One of the other things that kind of stood out from when I was looking at your abstract was using potential chemical elicitors
Yes.
um, to kind of, I guess, mitigate some of the negative effects of HLVd. So please tell us more about that.
Yes. So chemical elicitors or synthetic elicitors, also called plant growth regulators, as the name suggests, it's a chemical that mimics plant hormones. So it's essentially giving the plants a little boost to get their defenses going. And this is not, I didn't come up with this idea. Uh, we've been using it in plant pathology and in entomology research as a way to trigger plant defenses, give them a little boost to get it activated. You could also think of it as a way to prime the plants,
Hmm.
and it can protect against diseases and pathogens because naturally plant hormones, or what we call phytohormones, are doing this, but they're doing it at a low level so that the plant can um, devote all the resources to the different functions. We want to give it this boost so it can get activated. And the reason that I was interested in this research is because cannabis and hemp can be considered organic production systems, right? Because the repertoire of insecticides and pesticides are so limited.
Right.
All we have, and so there are only a handful of products. We don't even know their efficacy. And it turns out that these chemical elicitors are actually registered for use. So that's one of the reasons we were interested in this. And so our idea was to just boost the plant's inherent immunity to better defend the viroid.
And so was this prior to infection?
No, we didn't do it prior. So we infected the plant, and a week later, so we got the plant going with the infection, and then a week later, we sprayed these elicitors, and then sprayed every week for up to 3 weeks. And then subsequently, I think at the 4th or the 5th week, we took tissue and analyzed for HLVd levels using qPCR, and then let the plants go to flowering, and then did the cannabinoid analysis. So we debated, should we do it spray first, then infect, or infect and then spray? We decided to do, and I decided to do the former, because so we got clones from infected plants, so they were already infected, ready to go. Because that's probably we figured that's probably more of the natural situation. The plants are already infected.
Excellent. And so what were the results?
Oh my goodness. So you know this already, and this is how it goes with research. It's never, yes, perfect result. Always more questions to be asked.
Trade-offs, right?
Yes, exactly. But it's great. We did 2 rounds of it, and we're actually getting ready to do a 3rd round. So the results were mixed bag, but very interesting. What we found was, yes, indeed, a certain phytohormones or chemical elicitors reduced HLVd levels. Interestingly, they also increased the biomass, the yield of the plant, which we were excited about. But go to look at the flower production and cannabinoid levels, and it was, the major ones like CBD and THC was actually reduced.
How much?
I don't remember I could pull it up. I'll tell you biomass, like almost 50% for salicylic acid. The biomass
increased 50%.
Yeah.
Compared to an infected plant?
Yeah. No, they're all infected so the control was ethanol, because that's the solvent salicylic acid dissolved in. In ethanol, the biomass per plant was 8.3 grams. And in the salicylic acid sprayed, it was like 15%, 15 grams.
Oh, wow.
Yeah.
So almost double.
Almost double. Because these phytohormones also cause the plants to cause cell division, delay senescence, and essentially giving it better overall health.
Excellent. So
I'm just picking salicylic acid. They all look pretty much the same. Some of the other ones I'm saying, brasinolides was 12.8 grams compared to 8. So a little less than 50. So let's look at cannabinoid. We can again pick salicylic acid. Okay, so THC or THCA was 1900 microgram per gram in the control in ethanol. And in SA, it was 1300 microgram per gram. What is that math? 30%.
How much?
30.
30, right.
30%.
So basically 100% increase in biomass.
Yeah.
But a 30% decrease in THC.
Yes, in THCA.
Interesting.
Yeah. But not so interesting, because guess what? Just after we did this, this was a 2-year study from 2022, and he graduated in fall. In fall, here comes a really nice study, and I can talk about that, showing that in fact, higher levels. So our goal was to cause induction. The aim of our study, because we were kind of going blind, was we want to make sure these pathways in the plant, salicylic acid or what have you, was turned on, So we went a little high on the dosage, and then a paper came out saying higher dosage can reduce cannabinoids, whereas a lower dosage.
Mm.
So since that paper came out, we've got to fine tune the rates. It's not so over applying it can improve reduce HLVd levels and biomass, but does not have the same impact for cannabinoids, which is important. So you've got to fine tune this, and so we're redoing that experiment. It's already started, and I should have some new data for CannMed.
Excellent. So, so there is some fine tuning in figuring that out, but some
There is
potential there.
potential. Yes, for sure there is potential. For instance, we applied 10 millimolar of salicylic acid, and the papers are saying 1.5 is probably the better value, better concentration to go. So we may not see an induction in a PCR gel of the salicylic acid pathway marker gene, but that's the way to go. So we're going to play around with dosage and timing to fine tune that, because growers don't have too many options for these for HLVd. And the other nice thing about these elicitors is they also confer protection against pests. My own lab has shown like salicylic acid can reduce thrips,
Hmm.
right? So you are literally, and jasmonic acid are good against aphids. So you can actually kill two birds with one stone. So it's just a matter of fine tuning.
Great. And now, in terms of accessibility to these compounds, these chemicals, whatever we're calling them, elicitors,
And
are they easily accessible, pretty affordable as well?
yes, pretty affordable. Not all. So we used, like chemicals, standard grade chemicals, because this was a scientific study, but these products are available in the market to buy and apply. Some of
Excellent.
them are commercially available. And actually, for this new round of experiments we've got going on, I've asked my lab manager to try to get the commercially available ones. So we try to make it as applicable to growers.
Excellent. All right. So, in terms of practical advice to growers who are concerned about HLVd, potentially dealing with HLVd, based on the studies you've done and the research you've done, what are some sort of practical strategies to prevent or eliminate an
Mhm.
HLVd infection?
So we are finding that plant health really is crucial in terms of our field experiment, where we saw that healthy plants were impossible to even get infected. So that was key. And then these elicitors that boosted. Well, there were these trade-offs. Um, you know, um, Keeping the plants healthy did see reduction in the viroid levels. And given that now we know that these insect pests are vectors, I think monitoring for both early symptoms of the disease, because it just takes one plant to serve as a host for secondary spread, right? It's not
Mhm.
just you quarantine it, but it's maybe it may be too late. The insects already have it, and then they are going around infecting it. So I think monitoring for insects. So we have sticky trap, and we do this in my lab. It's the bane of my lab too. We've never had this much quarantining and care. Everything is in different places, and I know growers are already doing this. So keep your replace sticky cards often, and testing, which I know is expensive. And we actually have a project underway to develop some on-site um, field-based diagnostics, because it's even 20, what is it right now? I think it's $25 per sample. It's so
Yeah.
expensive and time consuming. So I think, uh, yeah, I know there
Yeah.
are some tests out there, but we are trying to work something out with some collaborators in University of Wisconsin, Madison. So I would say monitoring, sanitation, avoid cross-contamination, and testing based on the research that I know. I hope in the coming years, some of the things we've talked about, breeding for that they would be resistant variety genotypes, varieties, and genetic engineering. I mean, USDA approved these transgenic hemp, right? At least in theory.
Mm.
So I think those are all possibilities. We'll have to think about the sort of social stigma of GMOs that still exists a
Right.
little.
Yeah, of course. And, interesting thing that we're adding to this year's CannMed, we're having a whole genomics workshop all
Right.
about, it's kind of a half-day workshop exploring the use of genomics in breeding and in different applications for cannabis. So we're going to be talking about, genetic engineering and GMOs and touching on those things. So I think it's going to be a great session to sort of have those conversations, because it seems like that's the way things are eventually going to go.
Going to go, exactly. No, I saw that. I'm super excited about it. I saw Todd Michael is coming.
Yes.
Yes, so I'm very excited. Yeah,
Excellent.
so this one, because it's so resilient and so,
No,
we
and I
need
was
we need all throw the whole kitchen sink this approach for it. Sorry, Ben.
absolutely. No,
Yeah.
and no, when you were talking about plant health, and it kind of just clicked in my head, and as I understand it, outdoor hemp is largely grown from seed,
Yes,
whereas indoor
yes,
cannabis largely grown from
yes.
from
Clone.
clones. And there's
Correct.
always been the conversation of, growing from seed that have more vigor and are
Yeah.
just more resilient plants. And I wonder if that could be, part of why you're seeing more resilience in the hemp plants.
For sure. So in our field study, we had a what we call a dual purpose grain fiber hemp variety, which was grown from seed, and we had a high CBD line that was clonally propagated. So we tried to do both,
Mm,
but it didn't matter. Both
interesting.
dual the seed grown and the clonally propagated high CBD line that we like to use in our lab did not have
Hmm.
any difference in the infection rate. No, but this is just one clone, right?
Yep.
But that's not the case in the cannabis industry. So I think there's something there about the plant health and the conditions.
Excellent. All right. Well, Punya, winding down here, I did want to give you an opportunity to share any other resources or links that you think might be useful for anyone who's watching or listening who wants to learn more about this topic or your work. Please go ahead and share.
Oh, this is perfect because my team is putting together hemp entomology website through my lab website. It's almost ready to go. It's essentially a curation of all the reliable resources for pests and diseases, including some of our latest research. One thing we're planning to do is have a newsletter to growers, both cannabis and hemp. We're going to put that out through, this is the Colorado Department of Agriculture newsletter. And so we get all the hemp registrants, but I would love to reach the cannabis industry so they can, have more interaction with the university folks who are actually doing this research with, for the betterment of the growers, for the scientific and intellectual merit of it. So once we are still finalizing some aspects of this website, but it's essentially a way to just disseminate our, the current knowledge, get it out to the growers. I would love to send it to you and maybe in this talk have it there so growers can sign up for all the latest research in my lab and, other collaborators' lab, so they don't have to go reading scientific papers. So I'm excited about that. We are continuing to work on the virus vector interaction, as I have already mentioned to you. The other interesting aspect is the seed transmission, which was probably very surprising
Mm.
to everybody, right? To find out that it's over 80% seed transmitted in hemp,
Oh, wow.
and not seed transmitted in hops. The the first host. So we are trying to look into that and understand the mechanism a little better. Because right now, I don't think any of the hemp is being, the seeds are being tested for, certification or what do you say, state to state transportation. So it could become a big issue if we don't do anything proactively.
Wow, you said 80%?
I think so. I'm pretty sure
Wow.
it's staggering in hemp. So I think having certified seeds, clean stocks, start clean would be the way to go. And so the lines we used in my field study, the high CBD that was clonally propagated came from a tissue culture lab. So we knew it did not have anything. And so with the seeds, we tested to make sure that they were in fact negative, but not to say that there are potentially hemp out there that has HLVd.
Excellent. Well, all right. Well, Punya, yes, please do share the link to that site. I'd be happy to put it in the show description so it's easy for folks to find. And thanks again for joining us on the podcast and sharing this information and giving us a nice preview of the information that you'll be sharing at CannMed 25 down in Puerto Rico.
Yes, I can't wait to meet you in person. I don't think we have. No,
I don't think so.
Yes, so that'd be great. And no, I really appreciate you putting this podcast together and really helping the growers out, which is the bottom line, right, for all of us. So
Absolutely.
I appreciate that.
Excellent. Well, thanks again, and we'll see you in June.
Yes, bye.